Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today. I'm your moderator, Victoria Rees, editor of Drug Target Review. Today's speaker will be Dr. Leighton Smith, who is the Executive Director and Head of Biology and Pharmacology at Sanford Burnham Prebys Medical Discovery Institute. Following his presentation, we'll be moving on to a live question and answer session where you can pose your questions to today's speaker. Please remember that you can submit your questions at any point during the webinar using the questions panel situated in the menu on the right. But without any further hesitation, may I pass over to today's keynote speaker, Dr. Leighton Smith.
Thank you, Victoria, and thank you for joining this webinar. Today, I will be introducing you an innovative forward trafficking approach to unlocking orphan GPCRs. And to demonstrate the utility of this approach, I'll be presenting a case study targeting GPR20 and 22. As an overview of today's webinar and a summary of learning objectives, we'll first start with the introduction of orphan GPCRs as drug targets and the challenges related to identifying lead molecules for these targets. I'll then be introducing an innovative LIFT platform specifically designed and deployed for orphan GPCR lead identification. As proof of concept, I'll show our discovery of SBI 6602, a potent agonist of GPR20. Recent data has allowed me to expand this story, and I'll show you a few examples of application of the LIFT technology to other orphan GPCRs. At the end, I'll give you a summary and there'll be plenty of time for questions. Orphan GPCRs are an untapped resource for novel drug targets. Before we can define what we mean by this untapped resource, we must first define what orphan GPCRs are. Orphan GPCRs are G protein coupled receptors whose ligands have yet to be identified, and for which there are no small molecule probes, tool compounds, or drugs that activate them in a therapeutic manner. As far back as 20 years ago, Andrew Hopkins defined the term the druggable genome. In his paper, he sought to define the space by which the number of total genes in the human genome may be bound by a small molecule therapeutic and thus drugged, hence the name the druggable genome. On the right-hand side of my screen, you can see one of the figures reproduced from his Nature Reviews drug discovery paper. In it, two clear trends are observed. The first is that the vast majority of marketed drugs target either enzymes or GPCRs. The second is that less than 1/4 of all of the available marketed drugs target other receptors. The purpose of Hopkins' work was to define the total druggable space based on the number of genes in the genome and demonstrate the breadth of potential drug discovery targets. Others, myself included, saw this data in a different light. Indeed, what it says to me is that we have been very successful at using small molecules to drug enzymes and GPCRs, which of course leads to the logical question of how many undrugged members of those classes exist. Focusing on GPCRs alone and looking at the breadth of the superfamily, one can see that there are still a number of opportunities for drug discovery. Shown in the left-hand pie graph, you can see a distribution of all of the GPCR members of that superfamily. The vast majority, almost half, are olfactory receptors and not considered to be therapeutic targets. Roughly 1/3, or about 30%, have known ligands, and another 6% of these targets have been drugged. That is to say, there is a small molecule that binds and activates or inactivates the receptor to elicit a therapeutic response. However, approximately 15% of this eminently druggable class of targets remains orphanized. That is to say, There is no known ligand or small molecule probe for that particular orphan receptor. At Sanford Burnham Previs, we believe that these orphans represent an untapped resource for novel drug targets. Given the success of GPCR-based drug discovery programs, one is led to ask, why is it that almost 150 GPCRs, which are demonstrated to be good drug targets, remain untapped? The answer to that question is both technical and programmatic. The technical issues spawn from the fundamental process by which drug discovery occurs. In the early stages, high throughput screening is the most common source to identify a lead molecule. When one considers orphan GPCRs, there are three major technical limitations to high throughput screening and deploying those technologies to these orphan GPCRs. First, orphan receptors have unknown pharmacology or intracellular signaling cascades. That leaves the individual scientist to not have a clue of what the assay to be developed should be. You don't know whether you should start with cyclic AMP, calcium flux, beta-arrestin recruitment, or other intracellular signaling assays. Importantly, there is no known positive control or tool compound. I, for one, am loath to pull the trigger on a 500,000, 1 million, or even 5,000 compound screen when I don't have sufficient positive controls to establish assay performance, plate consistency, variation across plates, etc. Finally, there is a total lack of reagents. In this day and age, we've all gotten very used to being able to clone by phone or purchase cell lines that are readily available for us to interrogate our target of interest. Because of the lack of research on orphan GPCRs, there is also a dearth of reagents, including stable cell lines, antibodies, etc. These technical limitations make it quite difficult to begin a high throughput screening campaign. At the programmatic level, orphan receptors suffer from little or no target validation. That is to say, there are very few studies in either mice, rats, other animal models, or humans that would lead us to understand the direct relationship between an orphan receptor and a particular pathology. Therefore, there is no value proposition from a drug discovery standpoint, nor is there a sufficient rationale for conducting deep, basic scientific research around a given target. We developed the Ligand-Induced Forward Trafficking Assay, or LIFT, as a solution to the technical problems of orphan GPCR screening. I'm going to spend a few minutes introducing this technology to you because it is a little different and requires a different in thinking to understand the data that I'm about to show you. On the right-hand side of the screen, you'll see an animated cartoon showing a small molecule that binds an orphan receptor target that is retained in the ER. This ER retention is achieved by the use of specific point mutations which have modest changes to the tertiary structure of the target and lead to ER retention through a failure of this mutated receptor to pass through ERQC. In the presence of a small molecule that physically interacts with the receptor and binds to it, a conformational shift occurs, allowing for the ERQC machinery to approve this mutated receptor and transport it through the Golgi, ultimately to the plasma membrane. We have tagged the mutated receptor with what's known as the PK tag, or the prolink portion of beta-galactosidase, and used beta-galactosidase enzyme fragment complementation strategy to report the translocation of the receptor from the ER to the early endosome, as shown in this cartoon. Upon translocation from the ER through the Golgi to the plasma membrane and ultimately to the early endosome, the activated receptor binds the prolink tag to the remainder of the beta-galactosidase enzyme, known as the EA tag, forming a complete functional beta-galactosidase enzyme. Addition of substrate allows for the production of light and the measurement of the signal in a standard plate reader. This is the DiscoverX beta-arrestin recruitment and path hunter based EFC technology that is well known in the high throughput screening world. This approach has been applicable to all Class A GPCRs that we've studied. Importantly, it addresses the 3 technical limitations I discussed in the last slide. There is no need for a priori knowledge of receptor pharmacology. You notice I'm not proposing a second messenger assay here, which would require me to know what second measure I should measure. In addition, because of the nature of our ER retention and the subsequent ER-QC machinery, there is no need for a known ligand of this particular receptor because this affords a universal positive control in the form of the proteasome inhibitor bortezomib. It's important to note that bortezomib is not an agonist or antagonist or any other form of ligand of the target receptor, but rather by inhibiting the proteasome, bortezomib reduces the quality control stringency of the ER and allows for enough of the receptor to translocate regardless of its status of bound or unbound to a small molecule. Collectively, the LIFT assay provides us with a homogeneous cell-based format suitable for HTS and available to interrogate large chemical compound libraries for the identification of small molecule ligands of orphan receptors. As proof of concept, we took the orphan receptor GPR20 and applied a LIFT assay to the identification of small molecule probes for this orphan receptor. However, LIFT alone is not sufficient to produce a validated hit, nor is it sufficient to support drug discovery efforts downstream. Indeed, LIFT is the beginning and perhaps the center of a comprehensive repertoire of technologies for GPCR drug discovery that is needed to not only screen a large compound library, but ultimately validate the compounds and advance them to therapeutic discovery. At Sanford Burnham Prebys, we start with the LIFT assay and combine an entire repertoire of technologies that are currently available, including traditional technologies like second messenger assays, the hit hunter for cyclic AMP, and additional assays for beta-arrestin recruitment, receptor internalization, but also label-free approaches for cellular impedance, competition radio receptor binding assays, as well as phenotypic and high content screening imaging. Applying this repertoire of technologies to GPR20 led us to identify the first small molecule probe of this orphan receptor. At this point, I will be presenting our screen and our results of that screen, as well as hit validation. Having established the LIFT technology, we applied it to GPR20. And now I'm going to show you some of those exciting results. Before I show you the results of our screen, I need to rewind a little bit and set the stage for why we might be targeting GPR20 as a drug discovery project. To do so, I'm going to give you just the smallest amount of information regarding the physiology of gastric motility and the regulation of the pacemaker function that regulates gastric motility. Interstitial cells of Cajal, or ICCs, are spontaneous pacemaker current generating neurons which reside in the smooth muscle plexus of the gut. They're responsible for generating slow wave peristaltic action that causes mixing of food in the stomach and ultimately the progression of food through the intestinal tract. Loss of ICC function and population density is associated with a number of gastric diseases, most notably diabetic gastroparesis. This can manifest as tachygastrias, in which food moves very quickly through the small intestine, or bradygastrias, in which food fails to move through the small intestine and large intestine at an appropriate rate for absorption. On the right-hand side of this is a cartoon in which the interstitial cells of Cajal, acting as interneurons, transmit impulses to smooth muscle cells, in which leading to peristaltic flow in the intestine. In the absence of ICC, patients with diabetic gastroparesis or other gastric disorders suffer from a tremendous amount of gastrointestinal distress and ultimately may necessarily be hospitalized for surgical resection of that section of the intestine. Of interest to the gastroparesis therapeutic discovery is that GPR20 is the most abundant transcript in ICC. Shown here in this table is a series of RNA-seq and Affymetric array studies looking at a variety of ICC derived from both human and mouse intestine in various different areas of the intestine. In the top yellow highlighted portion of this table, you can see that GPR20 is one of the most prominently expressed transcripts in ICC, leading us to conclude two things. One, GPR20, like KIT, shown in the columns to the right, is a molecular marker for ICC. And number two, GPR20 likely plays a role in the regulation of ICC function. In order to test this hypothesis, we would need to determine the role of GPR20 in cells and animals, and the best way to do that from a therapeutic discovery standpoint would be with a small molecule probe. Hence, we developed a lift assay to identify small molecule probes of GPR20. Here's a general overview of our Orphan GPCR lead discovery strategy, which is applicable to GPR20. And this includes lift assay development, as well as secondary messenger assays for GPR20 in particular, cyclic AMP and beta-arrestin. It includes high throughput screening of our compound collection at the Institute, hit confirmation in both the primary lift assay, a counter screen using the same lift background, that I'll talk a little bit more about, and then ultimately a wild type GPR20 assay. Finally, at the bottom of this testing funnel, we include relevant iPSC-derived neurons or human cells, in this case ICC cells, as a final step for validating the pharmacology and the utility of the probe. It's important to mention that while the LIFT platform uses the Eurofins DiscoverX enzyme fragment complementation strategy at the beginning of the screen and as a primary approach, this is a highly artificial system. The mutations we induce in the target receptor are not related to human disease. They exist solely to facilitate receptor retention in the ER. Therefore, it's important to move very quickly to wild type assays using platforms like the the cyclic AMP assay, Hit Hunter, and beta-arrestin Path Hunter assays. Finally, given the orphan nature of GPR20 and other targets, it's essential to do broad selectivity panel analysis. For this purpose, we've been working with Eurofins DiscoverX to use their GPCR Max panel to make sure that our compounds are targeting GPR20 or our other receptors of interest and not other orphans. The specific testing funnel for GPR20 is shown here. Again, you'll see that we start with lift and we move rapidly through a series of stage gates, sorting out the hits, validating the hits, and in the end, we move quickly into a wild type GPCR format using a traditional assay. It's in here that we will conduct our SAR and probe development activities prior to moving on to validation studies. Assay development proceeded as one might expect. The first step was to generate the ER retention clones and validate that by immunocytochemistry. Shown here on the left panel, you can see that the GPR20 clone tagged with the PK and probed with an alexafluoridide antibody showed here in green, and the ER dye showed red. You get good co-localization of the target of interest. We can measure the overall expression of GPR20 by lysing the cells and adding exogenous enzyme fragment complementation subform EA tag, and see that there's an increase in the luminescence in the presence of exogenous EA and GPR20 cells compared to parental cells. Cell seeding density, DMSO tolerance, all were optimized for screening in 1536 well plates. And importantly, we generated a concentration response curve of the universal positive control bortezomib. Here is the results of the LIFT primary UHTS ultra high throughput screen of the Sanford Burnham collection for activity at GPR20. Shown here is the histogram of the hits, showing a nice spread between the red positive control bortezomib and the blue compounds that were tested. Yellow, it represents DMSO controls. The inset shows the scatter plot of the hits, and the assay performed quite well. You can see that we have a very strong signal from our positive controls, a solid background of 7.2, and an assay window of 5.4. Importantly, a Z-factor analysis revealed a score of 0.57, and a robust Z-factor corrected with gene data gives us a score of 0.55. Minimum threshold for performance of 0.5, so the LIFT assay is highly amenable and performs well in a UHTS scenario. We tested compounds at 10 micromolar and observed approximately 390 hits, which constitutes a 0.3% hit rate. SBI 6630 emerged as the validated hit agonist of GPR20 from this screen. Hit reconfirmation was performed using the GPR20 LIFT assay, in which you can see here in green, we have a nice concentration response curve in the presence of GPR20. We use a LIFT assay, the same technology presented for GPR20, but targeting the beta-2 adrenergic receptor, ADRB2, for selectivity. It's important to note that this is the same reporter system and the same background cell line. So you can see here that 6630 has no effect on ADRB2, indicating a high degree of selectivity for GPR20. As I told you before, it's important to move away from the artificial aspects of LIFT once you've identified clone compounds that are active at that clone, and move into a wild type scenario. In the bottom panel, you can see the effects of 6630 on wild type GPR20 assessed using the Hit Hunter cyclic AMP assay. Again, you have a nice concentration response curve, and that effect is specific to cells expressing GPR20. Shown in orange, you can see the effect of 6630 on cyclic AMP in parental cells lacking the GPR20 receptor. While I can't show you the full structure, I can show you a portion of the structure to give you an idea that this is a reasonably small molecule, and it is a drug-like molecule meeting many of Lipinski's rule of 5 and other physicochemical limitations and parameters that give us confidence of being a highly drug-like molecule. We next sought to determine whether 6630 physically interacted with GPR20. Using backscattering interferometry, we assessed the ability of 6630 to physically interact with GPR20 in membranes from cells expressing the receptor compared to membranes not. Shown in the dark squares is the change in mean polarizability that is detected by backscattering interferometry in the presence of increasing concentrations of 6630. In contrast, in the absence of the protein GPR20 and in membranes from parental cells, there is no change as shown in the dark triangles. In the backscattering interferometry signal, of mean polarizability in the presence of increasing concentration of compound up to 10 micromolar. The specific signal is shown in the dashed line and the open circles. Using this saturation binding isotherm from the backscattering interferometry study, we were able to calculate a KD of approximately 13 nanomolar. This data shows that not only was the GPR20 lift assay suitable for screening, but identified a drug-like small molecule lead that was active at wild-type GPR20 with a high degree of selectivity and physically interacted with our GPR20 protein. This meets our criteria for a validated hit. It is important to validate lift-derived hits in native contexts. To do that, we turn to the human cell line GIST, a gastrointestinal stromal tumor cell line derived from human ICCs. We first use Western blotting and fluorescent antibodies to confirm the expression of GPR20 and GIST, and this particular Western blot data is shown in the center of your screen here. Using these native human cells, we utilized a cellular impedance system to measure the change in cellular impedance in response to 6630. Addition of 6630 at a variety of concentrations alters the cellular impedance in the manner shown in the left-hand raw data trace. The peak change in cellular index in response to compound can be plotted in a concentration response curve, as on the right, and one can see that 6630 leads to a concentration-dependent increase in cellular index changes in human GIST cells, telling us that 6630 is active in a human cellular context that's relevant to GPR20 drug discovery. Consistent with literature indicating that GPR20 signals through cyclic AMP in a G-alpha S manner, it is sensitive to pertussis toxin and to cholera-toxin. However, other inhibitors of intracellular signaling pathways have no effect. All molecules emerging from lift for GPR20 or other targets should demonstrate SAR. We have identified approximately 90 compounds that were commercially available and employing substitution across 4 regions with demonstrated ranges of activity. Moving from the 6630 compound through this limited SAR expansion, we've observed a 10 times improvement in potency. We maintained a high degree of selectivity over SSTR4, which is the closely related analog of GPR20 based on amino acid sequence, as well as parental cells. This is still a very early lead discovery project, so poor stability in plasma continues to be an issue. Core substitution and additional solubilizing functional groups are ongoing chemistry efforts, and this is happening on regions R1, R2, and R3. I show you this data to further demonstrate the utility of lift for finding potential lead molecules. Using an advanced analog of 6630, the 6602 molecule, we moved into a more thorough characterization of the role of GPR20 and GIST cell pacemaker function. You'll recall that I told you that the GIST cells are a model of ICC, and the primary function of ICC is to control the gastroparesis and peristaltic motion in the intestine. In vitro, one can monitor the pacemaker function of GIST cells. Using optogenetic reporters, we can demonstrate a dose-dependent response to 6602, measuring calcium flux using the CG-AMP5 fluorescence reporter. An example of this data is shown in the trace of photographs at the top of the right-hand corner. As I mentioned before, ICC controlled the pacemaker function of the gut. This can be observed using these reporters as spikes in calcium flux, shown here in black under control conditions. The calcium ionophore ionomycin ablates this intrinsic spontaneous pacemaker function of the GIST cells. Treatment of forskolin affords one a chemical model by which one can induce a dysrhythmia in GIST cells, much like you would see in the native context in an intact intestine. Application of forskolin, shown here in red, eliminates the rhythmic pacemaker function of the GIST cells. Application of the 6602 molecule restores the rhythmic function of these pacemaker process in GIST cells, and that's shown in blue versus red, and is more similar to the black trace in the absence of the forskolin. While this is very preliminary data, we believe that the use of 6602 meets two requirements. First, it validates GPR20 as a functional regulator of ICC pacemaker function. And number two, it further validates that activators of GPR20, like 602, may be therapeutic molecules for the treatment of gastroparesis. Now that I've shown you that 6602 activates ICC pacemaker function, it's important to demonstrate that this is not an off-target effect. Using the Eurofins DiscoverX GPCR Max, as well as other safety profiling panels, we tested 6602 at a concentration of 10 micromolar against 241 GPCRs, 19 nuclear hormone receptors, and 19 receptor tyrosine kinases. A radial plot of that data is shown here on the right. As you can see in green, the activity at GPR20 is quite high, giving us a very strong signal. There are other activities shown in the bullseye with modest activities at 1, 2, 3 members of the chemokine receptor family. Because of the potency of 6602 at GPR20 and the high concentration in which we've tested 6602 in this panel, we do not believe that these modest effects of 20 to 30% activity reflect a significant off-target activity of 6602. What's interesting about the LIFT assay is, unlike a classic second messenger or beta-arrestin recruitment assay, LIFT hits comprise multiple scaffolds as well as mechanisms of action. I didn't have time to present data on the 3656 or the 5772 scaffolds, but these compounds were found with LIFT, were also highly selective for GPR20, but showed differential activations and differential pharmacological activities when examined in both wild-type GPR20 overexpression cells and ICC cells. This is because the nature of LIFT is to identify binders of the receptor rather than functional compounds, providing a unique and likely more rich hit set than a traditional second messenger or beta-arrestin recruitment assay would do alone. One question you might have, now that I've shown you our discovery of GPR20 agonists and the applicability of LIFT, is, does it apply to other orphan receptors? And the answer is yes. As part of a National Institutes of Mental Health sponsored program, we are applying LIFT to 10 orphan GPCRs and have recently completed work on GPR22 and GPR88. I won't go through the entire testing funnel for GPR22. I'll just spend a few moments sharing key results from that effort. The testing funnel is very similar to what you saw for GPR20, where we start with a LIFT assay and move as quickly as possible through to a wild type assay and subsequently into functional assays. The paradigm starts with LIFT and involves both, in this case, the HID Hunter cyclic AMP and the PathHunter beta-arrestin assay, as well as the GPCR Max panel presented just a few slides ago. As with GPR20, GPR22 performed well in a lift assay using a slightly smaller collection of compounds, but nonetheless generating a strong signal to background and Z-score. We identified SBI 2534 as a validated hit. Again, first looking at the activity in a concentration response mode of GPR22 lift, and selectivity was tested by ADRB2 lift. As I've said before, with GPR20 and 22, we moved rapidly into a wild type scenario. And you can see here in green a concentration response curve of 2534 at the wild type GPR22 versus parental cells shown in triangles. Unfortunately, 2534 compound is poorly soluble, so we're not able to generate the full plateau of the curve that most pharmacologists would like, but you can see that we're getting a nice concentration response curve in both assays despite solubility issues. What's most interesting is that 6274, an analog of 2534 that was identified through an analog by catalog approach to expanded SAR, actually showed inotropic effect in human iPSC-derived cardiac myocytes. I don't have time to go into the physiology of GPR22 and the regulation of cardiac inotropy, but suffice to say that GPR22 has emerged as a potential novel target for heart failure because of its effect on cardiac function in mice. This data shows that the compound 6274 has a potent ability to increase the inotropy, that is the contractility of iPSC-derived cardiac myocytes, almost as much as the control compound isoproterenol. Vehicle and control compounds had no effect. Interestingly, the rate at which 6274 stimulated the increases in cardiac contractility, in this case iPSC-derived cardiac myocyte contractility, was flat, indicating that there is a direct effect on contractile physiology rather than rhythm. That is to say that activators of GPR22, like 6274, stimulate an increase in the contractile force of the heart rather than the rate at which it contracts, suggesting that this particular target may be novel for the treatment of cardiovascular diseases, including heart failure, because of the unique effects on inotropy versus chronotropy. My second example of a broad applicability of lift to other orphan GPCRs is GPR88. Again, this is our testing funnel. I won't go through it because you've seen it before. Here's the results of our high throughput screen. Here we screened 320,000 compounds, and we identified many actives that show selectivity for GPR88. What's most interesting about this particular example is there are a number of known scaffolds that have been shown to show agonist activity at GPR88, namely the PCAA compound and an RTI analog 13951, commonly referred to as RTI33. We've synthesized these compounds in-house and tested them in a GPR88 beta-arrestin recruitment assay. Neither PCAA or RTI33 showed an effect on beta-arrestin recruitment to GPR88 using the PathHunter cell line. In contrast, two hits emerging from our GPR88 lift screen show modest but dose-dependent agonist activity to recruit beta-arrestin at GPR88, and those are shown here in green and brown. As before, I cannot show you the full structures of these molecules, but I've shown portions that show you that demonstrate the difference between the established small molecule activators of GPR88 as well, and our compound collection. It's interesting to note that RTI33 and PCAA have effect on G protein signaling through GPR88, but not beta-arrestin signaling. We are currently studying the effects of 9094 and 9474. Excuse me, I misread that. The two compounds shown on the right in cyclic AMP. Nonetheless, GPR88 lift identified unique chemical scaffolds distinct from those established in the literature with unique pharmacological activity at 88. In summary, I'd like to close by demonstrating that we successfully developed novel list assay formats for GPR20, GPR22, and 28. We've completed high throughput screening campaigns from 50,000 compounds to almost 500,000 compounds using this platform. We are continuing to expand the number of assays and targets that are enabled by LIFT. As GPR6, GPR52, and 151 have assays developed and are HTS ready. Those full deck screens will be triggered in this quarter. I demonstrated that we identified several active hits at GPR20, and I mentioned that there were 4 scaffolds, each exhibiting different molecular mechanisms of actions, indicating that GPR20 lift is able to find a breadth of chemistry where traditional cyclic AMP or beta-arrestin or other GPCR assays were not able to do so. Initial studies using human cells revealed a role for GPR20 in the regulation of interstitial cells of Cajal pacemaker function, and that would not have been possible had we not been enabled by LIFT to identify the 6630 scaffold. Moreover, initial SAR demonstrates chemical tractability and improvements in potency of the 6630, giving us a strong level of confidence that that particular scaffold is a true GPR20 agonist and has the potential to become the first-in-class GPR20 regulator for the potential treatment of gastrointestinal motility disorders. I also showed you that GPR22 was capable of identifying hits in LIFT, and that a expansion of that initial hit SAR identified a GPR22 agonist with inotropic activity in human iPSC-derived cardiac myocytes, further validating GPR22 as a potential drug discovery target for heart failure. Finally, GPR88 LIFT identified novel chemical matter that was distinct from known GPR88 leads. GPR20, 22, and 28, GPR20, 22, and 88 continue to be pursued, while an additional 5 orphan targets are going to be screened this year. Our current focus is on GPR6, 52, and 151. I hope from this webinar that you have come to the understanding that orphan GPCRs represent an untapped resource for novel drug discovery, and that the challenge to orphan GPCR drug discovery can be both technical and pragmatic. While I can't validate every human target, and I can't create a value proposition for every orphan GPCR, I believe that LIFT is a unique pharmacochaperone-based approach suitable for screening large compound libraries that is demonstrable in its ability to identify novel probes and ligands of orphan GPCRs, and therefore meets many of the technical limitations preventing an exploitation of this rich source of untapped targets. So far, LIFT has been applicable to all Class A GPCRs tested. It remains to be determined whether Class B or C or other members of the GPCR family are amenable to this particular approach. Importantly, LIFT is suitable for HTS, and using LIFT, it produces novel chemical matter compared head-to-head with established assay technologies, and is likely to provide unique chemical results from lead identification strategies. On the limitation side, it's important to recognize that LIFT is highly artificial. The mutations that we introduce into the target receptor to retain in the ER are not related to any particular human disease pathology. They exist solely to enable the separation of the EFC tags so that we can have a reporter system that's suitable for high throughput screening. Therefore, a complete testing funnel requires not only the use of wild type receptors in heterologous systems, but is best employed with endogenous receptor assays in human cellular contexts. I can't take credit for all of the work done. This is a large amount of effort conducted by the members of the Prebys Center at the Sanford Burnham Prebys Medical Discovery Institute, and has been a long-time collaboration with a number of individuals at Eurofins DiscoverX, originally started with Tom Werman, who's moved on from the company, but continuing now with Jane and Daniel. We've also worked on GPR20 with Thomas Ordog at the Mayo Clinic. Jake Isaacs performed the backscattering interferometry study at Molecular Sensing, demonstrating the physical interaction between 6630 and GPR20. And Kirsta Durney and Michael Granziano at Terra Biosystems performed the iPSC-derived cardiac myocyte studies on our GPR22 agonist 6374. Thank you very much for your time, and I'd be glad to take any questions.
Thank you, Dr. Smith, for your excellent presentation. We now have the pleasure to introduce our question and answer session, where Dr. Smith will answer all of your questions. Don't forget that you can still submit any questions that you have using the questions panel in the menu on the right. So let's take a look at the first question. So, Dr. Smith, do orphan GPCRs tend to have low affinity towards their ligands that may limit their characterization?
That's a good question. It's not one that I can answer for all orphan GPCRs. As I described in the webinar, very little is known about these particular targets, in part due to the dearth of limited reagents available to study them. So it's difficult to make a broad generalization. I think the bigger challenge with respect to the orphan, the identification of the ligands for the orphan receptors is that while they may be low affinity, that it is unknown whether they are present in the cellular milieu in which the receptor is being tested, creating a confounding effect by an endogenously produced ligand. This was the case with the Orphan GPCR88, in which it is a proton sensor, so hydrogen ions activated it within the context of the cell culture material. But generally speaking, we feel that the approach to identifying small molecule tool compounds would circumvent some of these issues of identifying the natural ligand. Indeed, ligand identification through traditional methods is extraordinarily laborious and time consuming, and that's important and essential work. But we believe that the identification of these chemical tools might accelerate illumination of these particular targets by more rapidly identifying tools that can be used to probe their function in pharmacology.
Fantastic, thank you so much. So next question for you now. First of all, this person says nice presentation. And the question is the lift assay restricted to small molecules, or would it also work with cell penetrant peptides?
Another great question. We have only used it for small molecules. It is necessary for the potential ligand to pass through the plasma membrane and physically interact with the receptor that is retained in the ER. If one were to engineer a cell permeable peptide, or perhaps work with some other mechanism by which one can introduce the peptide into the cell, then it might be amenable to it. But having not done that ourselves, I can't give you any definitive answer.
Great, thank you. So next question for you. Can you speak to the potential applicability or limitations of lift with regard to macrocyclic peptides?
As with the last question, since I haven't personally done it with my team, I can't say anything specific about the macrocyclic peptides. Depending on the physicochemical nature of those particular molecules, molecular weight, hydrophobicity, et cetera, they might be amenable to lift. The key question would be cellular permeability and the ability to physically interact with the receptor. If those two questions have been identified, the macrocyclic peptide should work just as well as any other cell permeable small molecule.
Brilliant, thank you. So Arrestin and KD46330 show NM potency in the cell line study. It seems it is UM. The question is it this because of receptor population density? If you need me to read that one again, I'm more than happy to.
No, that's quite all right. I think that the answer is yes. The key difference between the beta-arrestin recruitment, the LIFT assay, the cyclic AMP assay, is that we're using a engineered overexpressed cell line. So there's a tremendous amount of receptor density available to bind the ligand and report on interactions with the receptor and subsequent activation. In the GIST cell line, the actual receptor number is quite low. I don't have the radioligand binding data to show you or tell you about a receptor density in a Bmax format, but given the nature of endogenous GPCR expression, we are not surprised by the rightward shift in the potency curve from nanomolar to micromolar. Excuse me. And we likely consider that to be reflective of lower cell surface receptor density in the native context.
Fantastic, thank you so much. So what instrument did you use to measure backscatter interferometry?
We worked with a company that is unfortunately no longer in business called Molecular Sensing. They had an instrument called the TrueBind instrument. Generally, if you're interested in that technology, there are available instrumentation through secondary lab equipment sales, but we've struggled to find a replacement for that particular technology in this case. There's hopes that the technology and its assets from the company will be bought by another company who can bring it to commercialization beyond what Molecular Sensing was able to do. But beyond that, I do not know where one can use BSI at this stage.
Thank you. So the next question for you now is, why does the signal induced by SBI 62474 decrease at higher concentrations?
I'm glad you asked that question because I meant to actually mention that the 6274 compound has solubility issues above 7 or 8 micromolar. Very common for a high throughput screening hit. And we are certainly going to be working on improving solubility with that compound through the addition of solubilizing functional groups to the scaffold. But that's why you see that hook shape. That is not to eliminate the possibility of a bell-shaped concentration response curve based on future studies, but currently the solubility looks like to be the most primary culprit of that.
Brilliant, thank you. So what is the difference between GPR20 and GPR22 and how they work, and how is this deferred to GPR88?
GPR20 and 22 are named so because they were cloned through degenerate PCR priming strategy, in which Brian O'Dowd's group cloned 3 receptors. I believe they were related to the somatostatin receptor, GPR20, 21, and 22. So the numbers are just related to when they were cloned and subsequently described in the initial cloning in the late 90s. There are some interesting overlaps between GPR20 and 22 expression in the human body, but we don't believe that there is a physiological or necessarily a pharmacological relationship to those 2 receptors, other than the fact that they are orphan members of the GPCR superfamily. The same can be said for 88. The reason these particular targets were exploited or applied to the project was they are expressed in the brain in discrete regions of the brain that are known to regulate cognition, behavior, and emotion. And as part of our NIMH funded program, we restricted our focus to orphan GPCRs that were enriched in those regions of the brain. And so that's why we have selected them. I should mention that GPR88 is considered to be fairly well established as a potential drug target for mental health. But GPR20 and 22 are relatively unknown. At last assessment, I believe there were 4 manuscripts in PubMed referring to GPR20, and one of them is a GWAS study that doesn't really talk about anything with respect to the pharmacology of the receptor, and the other 3 are very early discovery work. So it's one of the joys and challenges of working in orphan receptors. There's almost nothing known about them, which is both exciting and challenging at the same time.
Definitely. So how are the ER retention mutations selected?
That's a great question. Generally, we're looking for conserved regions in the transmembrane domain that result in modest kinks in those particular helices. So for example, in GPR20, we mutated tryptophan 175 to an alanine, and that very subtle mutation was sufficient to lead to ER retention. And that's our goal. We want to disrupt the structure of the receptor as minimally as possible. In the case of GPR52, we're having to make multiple mutations throughout the receptor, but that's just happened to be the case of that receptor. So I can't give you a specific rubric about the exact way to do it. There is an approach, and one has to sort of go through a little bit of trial and error to get it to work.
Great, thank you. So what is the overall hit rate success with orphan GPCRs?
Generally, we're in the 0.5 to 1% hit rate. Some of that is by design. We don't want to have too many hits, so we generally accept a z-score of greater than 3 and or an activity of 80% of control. But we also don't want to end up with 3,000 hits. So we need to have a manageable hit set. So we're never going to accept more than 1% in our approach. Generally, we're in that slightly below or right at 0.5% hit rate. Now, we recently obtained a new library of compounds that have been selected specifically because of their 3-dimensional fingerprints and pharmacophore similarity to known small molecule GPCR ligands. And we expect our hit rate to go up when screening that particular collection. But we don't have enough data on the performance of that library to say whether or not we would improve the hit rate. But at this stage, as I said, about 0.5% is what we expect from these compounds, or sorry, this collection of compounds with the LIFT assay.
Brilliant, thank you. So how long does it take to screen 200,000 compounds on the LIFT platform? And what's the average timeline for the complete screening funnel?
So we've never done 200,000 compound slugs. We've done about 350, about 450, and 550 and 100. So the one number you want, I can't give you an exact number. In a fully automated mode, we can run the large collection at Sanford Burnham Prebys of just under 500,000 compounds in about 5 days of active screening. We generally have less than 20 plates to rescreen, so just give it another actual day. So 6 days for 500,000 compounds, maybe 3 days for 200,000. These do require a 16-hour overnight incubation in order to get a very robust signal to noise ratio and a good Z factor. That seems to be the consistent performance. We've tried shorter periods of incubation time, as well as longer, but 16 hours overnight seems to work best for our workflows.
Great, thank you. And just to quickly let our audience know that we've got a high number of questions coming through, so we're just going to go past the top of the hour slightly to try and fit in as many as possible. But next question for you now. Can the ER environment, reducing conditions, protein content, etc., affect the receptor-ligand interaction?
I don't know the answer to that, but I would suspect that there'd be some impact of the ability of the ligand to bind the receptor within the ER. Certainly, if there are different chemical conditions, as you said, under reducing conditions, you might have an ability to enhance or reduce the activity of the compound. But remember, lift is a primary screen. The strategy should be to cast the widest possible net in order to get the most chemical matter, and through your testing funnel, narrow it down to the more relevant wild type, quote unquote, normal GPCR expression in a cell line or other types of assays.
Fantastic, thank you. So what's the average number of GPCR mutants one might need to test to have a lift-compatible cell line? Are there any good tools or bioinformatics for this purpose?
Well, of the 6 we've done, the only one we've really run into problems with was GPR52. Luckily, we've been enabled by the recent crystal structure that was published, I think, in 2020. And that revealed that there is a self-inserting loop. And we hypothesized that the reason we're not getting ER retention is because the self-activating loop insertion results in a forward trafficking event, regardless of the ER retaining mutants that we put in. Generally speaking, 1 to 2 mutations is all that we need. And we focus mostly on conserved regions within the structural domains of the transmembranes that confers this retention. As I said, there's not a great rubric for doing it, but there are principles that are, can be applied based on looking at conserved regions across the various different targets. It probably take my own webinar to actually go through the details of that, but we currently use Genocopia. We synthesize several potential mutants in parallel, and then rapidly test them with the ICC approach, the immunocytochemistry approach that I showed you in the slides.
Perfect, thank you. So how do you select the retention mutant used for screening, and how many are tried during assay optimization stages?
Well, I think I should have had a seminar on the mutation selection. As I said before in the answer to the other questions, there's no clear rubric that you find that particular amino acid and mutate that. The best thing I can say is that we look through conserved regions throughout the TM domains, and we focus on those areas that are known to be consistent across all GPCRs. I think what's important is what we try to do is make the least number of mutations that are needed. In many cases, we're able to find a single mutation that can be changed, a single mutation that will lead to ER retention. And that's generally pretty straightforward. I think we're getting better at guessing where they're at, but at this stage, it is still trial and error. Generally speaking, we pick somewhere between 3 and 5 mutations to test. And again, we use the immunocytochemistry localization in the ER to see if that's retention is obtained with those mutants.
Thank you so much. What benefits would GPR22 agonist have against classical medicines for heart failure, beta blockers, for example?
Well, that remains to be seen. Our primary focus here was to identify a tool that could validate GPR22 as a potential therapeutic for cardiovascular disorders, in particular heart failure. Our initial studies suggest that there is an effect of GPR22 agonism that increases the rate, sorry, not the rate, the force of contraction, as I said earlier, inotropy, without increasing the rate, which would be unique in an inotropic therapeutic. Most of them result in both increased rate and increased contractility. However, I think what's exciting about this particular compound is with a little bit of chemistry, we can publish the compound and other scientists can begin to explore the role of GPR22 in the regulation of cardiac function and perhaps provide further validation for it as a target for heart failure.
Fantastic. What kind of control are you using for orphans?
I assume you're referring to the positive control. As I mentioned in the presentation, LIFT is amenable to HTS in part because of the universal positive control in the form of bortezomib. Bortezomib is a proteasome inhibitor, and so it kind of falls into the logic that by inhibiting the proteasome, you induce ER stress. As part of that increased ER stress, the ERQC machinery is overall reduced, and therefore there is an increased translocation of a mutated receptor from the ER to the plasma membrane and subsequently to the early endosome. At least that's how we think it works. And so we're able to use bortezomib in all of our LIFT assays.
Thank you. How are the mutants screened to finally choose the right mutant to go ahead?
So there's three-step process. The first, as I mentioned, is to examine ER retention in an ICC study using antibodies directed against the ProLink tag, which are commercially available from Eurofins DiscoverX, as well as with an ER dye. We've done this with both manual microscopy as well as high content imaging and automated cell screening. The second is to examine the overall receptor expression level, again using the platform available from Eurofins DiscoverX, in which we lyse the cells and we add exogenous EA tag, which forces the complementation of the EFC and gives you an indication of the available PK tags that are there because EA is added in excess, and you expect to see a certain increase in the overall signal when excess EA is added to cell lysate. And then the third is our bortezomib. If we can see a increase in the overall luminescent signal in the presence of bortezomib relative to DMSO vehicle, we feel like we have got the right retention, receptor expression level, and translocation effects. And that's all part of our assay development package that includes DMSO tolerance, FBS assessment, and all of the general processes one would go through before triggering a high throughput screening campaign. Generally speaking, on the LIFT assays we've done, the protocol turns out to be quite similar, although there tends to be a difference in bortezomib sensitivity, which we believe is related to the overall expression of the particular receptor. And although the receptors are all under the CMV promoter, we do have variable levels of expression of the receptors, which may or may not be related to the mutations that we have introduced. But nonetheless, the expression level is sufficient to generate a good assay with a high signal, low signal, and a robust Z factor.
Thank you so much. Do you also see a small effect of the identified hits on non-mutated receptors?
Well, I think we'd like to see a large effect on the wild type receptors. So part of our testing funnel is indeed using just LIFT for the primary assay and then moving very rapidly into a wild type system. That system can be a traditional second messenger assay or beta-arrestin recruitment, or we can use the cellular impedance approaches if the second messenger is an unknown, probe that pharmacology, determine the second messenger, and work backwards from there. But our goal is to see strong effects at the wild type receptor. That's part of our inclusion criteria for advancing compounds. And indeed, as I showed you for the 6630 molecule for GPR20, we see a nice concentration response curve in both the lift and wild type cyclic AMP and beta arrestin recruitment assays for GPR20, and as well in the GIST cells that express it natively, albeit at a slightly lower potency, likely due to receptor surface densities.
So for a GPCR with known ligands such as ADRB2, which percentage of ligands are not detected in the lift assay?
That's a great question. We published in Assay Development and Drug Technologies a couple of years ago the application of the lift to ADRB2 in the screening of the library of pharmacologically active compounds, Sigma's low-pack library, which is perfect for this experiment because it contains many beta-2 ligands. In that particular study, we identified all of the beta-2 ligands in the low-pack library and 2 false positives, which upon testing at wild type did not have any activity. So in the only experiment we've done to actually test that hypothesis, all of the known ligands that were available in the library were identified, and there was a slight false positive rate, but no negatives.
Great, thank you. So have you ever thought of using heat to increase or decrease stringency of this assay?
We've not. Our robotic systems allow for certainly the incubation of the compounds at 37 degrees and for the 16-hour incubation period, but we've not explored higher temperatures or for that matter, lower temperatures to improve or reduce the stringency. Remember, we're getting a fairly low hit rate of somewhere around 0.5%. So I don't know that increasing stringency at the first primary stage would be, consistent with our goals. We feel like using lift to screen a broad swath of chemistry and then using the power of the testing funnel we've developed to narrow that chemistry down is the best approach to get the most robust hit set that is most relevant to the target of interest.
Great, thank you so much. And why is the EA tag in the endosomes and not at the plasma membrane?
Well, I didn't go into it too much, but what we realized early on in collaboration with Eurofins DiscoverX was that while we could put the EA tag at the plasma membrane, that internalization to the early endosome afforded us the opportunity to separate compounds that are actively internalized versus passively internalized when we examine the effect of the compound in a concentration response mode. Why was this important? Well, we realized that we would be able to segregate compounds that are stimulating an active internalization. I'll call them agonists because we don't know that they're agonists at that stage. They just have an agonist profile. Whereas an antagonist would be subject to a more passive internalization. In practice, what this looks like is that you have a large delta in the Emax of an antagonist at the bottom and an agonist at the top. So compounds that are active or agonist-like would have a higher level of internalization and a greater luminescent signal than those that are more passively internalized, such as an antagonist. So that's the benefit of using the early endosome tag. One could also look at the plasma membrane and move on, but we found that this additional ability to segregate based on Emax at in the EEA tagged version of the system was more beneficial for our overall probe discovery efforts.
Brilliant, thank you so much. Unfortunately, that is all we have time for today. But thank you so much for sending in some fantastic questions there. And also thank you so much to Dr. Leighton Smith for his time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please do take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. But now, on behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.
Thank you.